So I won't be working from a computer because I was sitting taking notes all through yesterday and this morning, and I have a stack of them down there, and I had to try and synthesize something out of all the information that I was hearing constantly. First of all, I want to express thanks, a great vote of thanks for Philippe Sansonetti and to Brett Finlay for putting on this day-and-a-half event. I think it's a very, very exciting time that they have managed to create with a group of people coming together who normally would not have anything to do with each other. And we are slowly learning how to talk across disciplines, perhaps, I hope. It seems as though we are in some areas, learning from each other in ways which will create a bigger picture than merely that of the microbiome at the basic level. I think we still obviously have a very long way to go. I see this as a beginning, and I hope there will be more meetings like this in which we can figure out ahead of time what areas need to be concentrated on in a way that will bring much more of a synthesis, an open and obvious synthesis together between the work of anthropologists and microbiologists. I think that we are I won't say unfortunately, but I think we are not surprisingly still essentially in two very different domains. We use different methodologies, different approaches, all sorts of things, very basic things are not clear. It became evident to me today that some people in the room don't understand that the largest number of anthropologists are not biological anthropologists. They are cultural anthropologists, and by far the long way. You could bring biological anthropologists into this room. They would communicate with you with no trouble, and you would be delighted with them, and they would be delighted with you. But many people in this room are likely to have more trouble with those of us who are cultural anthropologists who ask some troubling questions. Our task has always been to cause trouble, and we get a kick out of it quite a lot of the time. So to go back, but the presentations really have been remarkable, and I'm sure everyone sitting in this room has learnt a huge amount. We have several questions that we opened up with certainly remain. I think that, I can't remember who it was, but somebody at the beginning said one of the basic questions is who or what begat who and when? We certainly haven't answered this question. It's one of these questions that are probably unanswerable, and we never will, but it's one of these questions that should always be on the table, and we keep coming back to reflect on it again and again. Several basic things came up, either directly in the talks or indirectly. One of them, for sure, being the question that we started out with the Anthropocene. We didn't bring up the Anthropocene explicitly, aside from in the first very important talk. But the reality of the Anthropocene is with us, and it's here to stay. And it leaves us with a situation in which it is clear that humans are indeed transforming the globe in a way that it has never been transformed before, and at a rate which is phenomenal. And one of the effects of that, which we haven't started to document, but we're beginning to get glimpses at through some of the presentations being made here, is that I think perhaps all of us, certainly very large swaths of people, are permanently out of sync with their environment. And there's no way in which the microbiome must be struggling a lot to constantly adjust and adapt to the exposures that are slaughtering us. And just walking around the cities of Europe with all the toxicity, one can feel in obvious ways the effects in terms of asthmatic attacks and things like that, but in more subtle ways, I think many of us are beginning to feel either that aging is much worse than I, for one, thought it was, or there's something else going on as well, which is much more subtle, that we need to start trying to get to grips with. We also, it was repeated in many, many ways without being stated explicitly, that microbes are indeed opportunistic travelers. And they are out for themselves, they're not out for us. They make use of us, and we think we make use of them in certain ways too, especially with respect to development of antibiotics and things like that. But there's no doubt in my mind that they are actually in charge, not merely in massive amounts of by huge numbers, but in many other ways. Their opportunism is more subtle than is ours, and we have a lot to learn about that. And another variable that we are confronted with, which we didn't bring up perhaps as much as I thought we might, is the whole question of global warming. It was mentioned, but not all that much. And this too, of course, is obviously due to human activity, and it too is a major contributor in transforming the microbial world. They inevitably are part and parcel of this whole transformation. I think the historical papers that were given, both those with deep history and with much more recent history present in them, made it crystal clear that microbes wreak both havoc and do good. And again in the papers, many presenters went out of their way to point out the good that is done by bacteria. This is one of the things that we certainly have to, when we talk to public audiences, is to get the public to recognize the positive side of microbial activity. It's something I know that we have been beginning to tackle and talk about, but so far I think there's not much of a dent on the public in general, at least not those people that I've recently come across. We are for sure in what I prefer these days to call the capitalocene, not just the Anthropocene, but it is capitalism that has driven us in this direction and will continue to drive in this direction. And as someone who's always been sort of a bit on the left end of the spectrum, I firmly believe that we need socio-economic changes, political changes, as much as we do others, to try to get some kind of grip on this. And we shouldn't forget that side of it. Okay, so I hadn't realized, for example, Marty's remarkable paper, and I know Marty's work rather well. I'm working together with Marty. We're on the phone every 3 weeks and so on. But I hadn't registered that the number of diseases have increased that dramatically just since World War II. I mean, that change is horrendous when one thinks about it. And we live in this naive assumption that actually the number of diseases have decreased because we've got antibiotics and we get to the doctor more quickly and so on. This is a paper that needs writing out fully if a long, thoughtful paper needs to be published. And again, I will take the chance to say right here, in case I forget it, that I do believe that we should not be just writing for ourselves, for academia, for the intellectual community. Some of us should learn how to write papers which can be put into magazines, can be, obviously, quite a number of us have given public presentations and media presentations, but I think we also need to learn how to communicate in everyday literature to reach a very, very wide number of people. We are confident that the message we have, the various messages we have, are important and need to be better known. In that case, it's our job to get them out there. To the biggest question of the moment right here is, have we indeed been able to integrate the papers, the numerous papers that presented remarkable findings on the microbiome with an anthropological approach and the relatively fewer numbers of papers that started out from an anthropological perspective and did a more conventional anthropological presentation. And I would say basically that we haven't succeeded in doing that. I think we are still essentially in two different domains. And I think at the next gathering of this kind, we need to explicitly work much harder ahead of time in perhaps pairing up microbiologists with social scientists and historians and philosophers ahead of time prior to the meeting, to really think through how one could present integrated presentations. I must say that again and again, through these the talks that we've had at this meeting, I was sitting there thinking, what about the social? What about the political? What about the daily life of these mothers and fathers and so on? And I was actually, just with the Pascal's talk, I was thinking about the head of WHO, Jim Kim's work on stunting, which I trust you know about. He is an MD, but he's also an anthropologist, trained at Harvard, and he has done work on stunting in, I think it's Peru, it may be Ecuador. And what he found, because he started out from a much more social approach, rather, he didn't deal with the microbiome. He probably barely knows about the microbiome. He went in wanting to discover why these children were stunted, and very quickly came to a conclusion that the problem was the poverty of the mothers and their inability, even when they were given packets by WHO, to distribute the food adequately. So he interpreted a whole stunting problem in that part of the world as he didn't even think about the microbiome. He's thinking much, much more about the actual social dimensions and political dimensions of these problems and how they need to be transformed. And I think we have to pull these things together. We absolutely have to. And as I was saying, I was sitting here again and again through the papers thinking, this is great work on the microbiome, but what about the social? If we forget it, then we've lost half of the potentially really stunning results. Okay, so I'm just going to, rather than run through each paper and say, you did this well and you didn't do that well, I've got a packet of notes, but I'm not going to do that. That's boring. And everyone did things well, so, right, with a few little blips, in my opinion, and it's just my opinion. So. But I want to make just some more general comments, and then I'll wrap up what has been a highly stimulating time. The philosopher of science, Evelyn Fox Keller, who I presume many people know of, she has argued for some time that the role of the genome has been turned on its head with knowledge about epigenetics. And I personally am currently very interested in epigenetics, and I think this needs to be linked to microbiome work. She feels that the genome has been transformed from an executive suite of directional instructions to an exquisitely sensitive system that enables cells to regulate gene expression in response to their immediate environment. We live now, and somebody mentioned this, with what Scott Gilbert has described as a reactive genome. And we're in a post-genomic era, at least many of us are, in which we think of the environment and environments as the driving forces for health and illness, for the process of aging and development. This is a massive change. It's something which I don't usually talk about paradigm shifts, but I think this is something pretty close to a paradigm shift for those that have taken this up. So the Barnes and Dupré, who some of you know, who work in Britain, they have argued that if genes are conceptualized in effect as real entities, then they must at least be understood as composite rather than as unitary, somewhat analogous, they say, to the solar system or a forest or a cell culture. So they argue that a dynamic epigenetic network with a life of its own, a context-dependent reactive system of which DNA is just one part, has been exposed. And my conclusion from this is that if we follow this, we're living in a world in which contingency has displaced determinism. The microbiome shows us in an infinite number of ways how determinism has gone. And as somebody I was talking to yesterday and we agreed that fuzziness is a word that we need to be using and not feel bad about. We cannot live in a world of rigor. This doesn't exist. 2 decades ago, the neurobiologist Stephen Rose, who no doubt some of you know, argued that we must be concerned above all with the dynamics of life, that is, with process, including the continuous interchange between organisms and their environments. He argued that what he calls our lifelines are constituted by life processes which generate our sense of self. And with all of these statements, this audience can integrate the microbiome into these statements. Rose insists that we're defined by our histories, at least as much as by our molecular constituents, and individual histories comprise lived experience in environments natural and social. So in order to better comprehend the effects on human well-being of human-made environments created in the Anthropocene, in addition to the research findings of epigeneticists and other scientists, it behoves us, I think, to pay attention to historical, socio-political, and environmental variables. And in addition, we add in the microbiome. The philosopher Russell Keat, 2 decades ago, argued that a good deal of time has been spent in the social sciences, in the social sciences and in the humanities, in discussing the distinctiveness of human beings, while at the same time holding to an assumption about the non-distinctiveness of the human body. This is something that Bruno Latour has tried to deal with and combat. Several of us in anthropology have likewise been always followed what Bruno had to say about these things. And it is, in my opinion, essential that we get under the skin and not just, as anthropologists, work on the skin. And this is one arena whereby we can link with the basic scientists and who are working on the microbiome. But more anthropologists need to be trained to think comfortably this way. Anthropologists have, particularly cultural anthropologists, have had a bad habit of totally black boxing the body interior, leaving it deliberately to biologists, nothing to do with us, and it's left this horrible gap between these two worlds. Okay, so I want to finish up with an illustrative example. I think actually maybe 2 short illustrative examples to try and demonstrate the direction in which I would like us to go as we proceed on into the future. And it's to do with the intergenerational transmission of toxins. Based on many years of fieldwork that commenced in 2003 in Hanoi, Vietnam, the Danish anthropologist Tina Gammeltoft documented the devastating effects on reproduction that persists more than 40 years after the war caused by the chemical defoliant Agent Orange. The Vietnam War lasted from 1962 until 1971, as you may well remember, during which time the US military conducted an aerial defoliation program that was part of a forced urbanization strategy designed to force peasants to leave the countryside, where they helped sustain the guerrillas and move to the cities dominated by US forces. During this time, nearly 20 million gallons of chemical herbicides and defoliants were sprayed upon Vietnam, Laos, and Cambodia, destroying all plant material in 2 days, and in some areas, toxic concentrations in soil and water became hundreds of times greater than the levels considered safe in the US. Agent Orange contains, of course, the highly toxic chemical dioxin, and this has long-lasting effects, not just on the environment, but on human tissue, and obviously on the microbiome. Gammeltoft documents a widespread fear about the so-called dioxin gene, widely believed by many people living in Vietnam today to be increasing in the population over time. It's estimated that at least 3 million citizens in Vietnam suffer from serious health problems due to exposure to defoliants, and the rate of severe congenital abnormalities in herbicide-exposed people is reckoned at 2.95% higher than unexposed individuals. And animal research has shown that following fetal exposure, dioxin reprograms epigenetic developmental processes, the effects of which may become manifest throughout life. Cases of third-generation Agent Orange victims have been reported in which individuals exposed during the war have produced apparently healthy children, but whose grandchildren are born severely disabled. Vietnam was given membership in the World Trade Organization in 2007, and one result of this was heightened concern by the Vietnamese government about the international visibility of the health of the population as a whole. And so they started using extensive ultrasonography to ensure the birth of healthy children, 8-9 times during a pregnancy. One result has been that ultrasound is now being used repeatedly on a daily basis in Vietnam. And then there are problems with abortion in this country, and people wait very, very late to go ahead with the pregnancy. When they realize there's going to be problems, they then want a very, very late termination. So Gammeltoft's kind of work, there's a range of severe illnesses, of course, associated with dioxin exposure, including deadly cancers, Parkinson's disease, and spina bifida, in addition to those specifically associated with pregnancy. And the US has never really taken responsibility for this situation. So this, I would say, is a kind of example of daily life that still is present in Vietnam. But I think it's a cautionary tale for those of you doing research into the microbiome, that when one goes into a community to do microbiome research, you really need to know the long-term historical context. You need to know what those people have been exposed to in one way and another by various toxins, farm children, people working in the far north of Canada. These toxins drift up to the Arctic. They end up in the bodies of Inuit. We need to appreciate this kind of global drift of negative compounds which need to be integrated into your kind of research. And just very quickly, a second example of what I call toxic local biologies, furnished by the mercury-contaminated Grassy Narrows Wogiboon River system in Ontario, Canada. The government claims that defilement of the river stopped 40 years ago when a paper mill was forcibly shut down after dumping about 9,000 kilograms of mercury into the downstream river. Today, mercury levels in the fish near Grassy Narrows are 15 times the safe consumption limit. And there have been lots of problems with pregnancies with women living near this area. The Grassy Narrows people have fought for more than 50 years for a cleanup of this river, and the Ontario Minister of Environment over the years has postponed it, put it off, and so on. And today what we have is 2 generations of people in Grassy Narrows, First Nations community, who exhibit symptoms of mercury poisoning, including loss of muscle coordination, numbness in the hands and feet, hearing loss, speech damage, and tunnel vision. And fetuses, of course, are particularly vulnerable to this kind of problem. We know a lot about mercury poisoning because of what happened in Japan years ago, and the Ontarians chose not to learn about the enormous problems that took place in Minamata Bay in Japan. So this is just another example of the kind of environments that you will be dealing with as you go to collect fecal materials to analyze the microbiome. I'm just giving a big cautionary shout here about making sure that the larger environment in all its dimensions, the history in all its long-term effects of history over the years, is integrated into your thinking as you try to interpret your findings. And I've stressed this because the one unhappiness that I had throughout the day and a half has been that again and again you presented fabulous data at the micro level about the microbiome, and the larger issues were just simply not addressed at all. And I'd like to see us really work on that next time. Thank you.